Good morning to everyone, and thank you for joining me for this presentation. I would like to start by also thanking the organizers for providing this opportunity for me to share some of the work that we have been developing at BioEasy Genomics and acknowledge that it is a great pleasure to us to finally become fully integrated members of the GenomePT infrastructure. So, the project I'm going to present today is born out of a collaboration between my group, the RNA and Systems Biology Lab at BioEasy, and the group of Raquel Chaves at the University of Trás-os-Montes, who is actually the PI of the project. So I hope that today's talk will do justice to the work of all the wonderful team involved. And this includes, of course, our joint PhD student, Mariana Lopes, the BioEasy Genomics core technician, Mariana Nascimento, who is now actually at the Max Planck Institute, and my colleague and facility co-coordinator, Ricardo Dias, but also our external collaborators at the Cambridge Comparative Genomic Resource Center and the Sanger Molecular Cytogenetics Core Facility. So, in addition to BioEasy Genomics entering the GenomePT infrastructure, this year is also quite exciting because we are celebrating the 20th anniversary of the publication of the first draft of the genome sequence, as you all know. Now, at its inception, the Human Genome Project was actually compared to our efforts in space exploration. And well, much like what we find in space, it is true that the genome has a lot of dark matter. And more to our interest, it does have some peculiar entities, which we call satellites. And as I'm pretty sure you all know, these parts of our genome were initially identified back in the 60s as DNA segments that would migrate as separate bands from the main genomic sequences in gradient centrifugation assays. We now know that, in fact, these satellites are part of the huge mass of repetitive sequences that make up the human genome, more specifically corresponding to the constitutive heterochromatin that is predominantly found in chromosomal telomeres, in centromeric regions, but also as interspersed sequence elements. Now, these satellites, they form really, really large tandem arrays, and they can be classified into different families depending on multiple aspects. For example, the size and sequence of their repeat unit, their relative abundance in the genome, whether they are present across all chromosomes or just predominate in some specific chromosomes, and also whether they are able to form higher order repeats. Now, these higher order repeats are basically stable arrangements of sequence variants of repeat units that will then repeat themselves across the large tandem array. In addition to their well-known structural and functional roles at the centromeres and telomeres, it's important to state that sat-DNAs play a very important role in genome evolution, acting as hotspots for chromosome rearrangements that can be linked to human and even to veterinary pathologies. Now, for example, the Robertsonian translocations involving the fusion of two acrocentric chromosomes with the loss of their short arms are in fact one of the most common chromosomal abnormalities found in mammals, and pericentromeric and centromeric DNA sequences are known to play a critical role in their establishment. For example, in domestic cattle, the translocation between chromosome 1 and chromosome 29 has long been considered a really serious problem by breeders, warranting the selective killing of animals that present this rearrangement to avoid what they believe will represent a decrease in fertility and overall fitness. So some of our team members have been for some time carefully mapping the structure of the rearranged centromere regarding the composition of satellite DNAs and comparing it to the structure of the original chromosomes. And they have really made some very, very exciting observations. Indeed, while part of the satellite's original sequences found in the individual chromosomes are preserved in the derivative chromosome, the fact is that the way the structure is organized is not what you would expect from a simple break and joining event. So, in fact, this final combination that is established here can only occur if a multi-step mechanism is in place that involves the joining of the two long chromosome arms, the inversion of an internal segment, and then the loss of extra satellite sequences, namely satellite 1 and satellite 1.273, so that in the end, this sequence is maintained as the functional centromere. So we have proposed this two-step model as an explanation for the actual process for the formation of this translocated chromosome. Now, for those interested in genome evolution, it is really quite striking to learn that this same translocation is present as a constitutive rearrangement in wild bovine species. So here's a tree of the bovine family, and our domestic cattle is here in the pink arm, whereas wild bovine display the translocated chromosome as their constitutive configuration. Okay, and Raquel Chaves has for some time been carefully mapping what these translocated chromosomes look like. And interestingly, the available evidence points to a very similar process of rearrangement, including the inversion and progressive loss of satellites to a point where the species that have diverged longest from the main branch only retain the critical satellite for centromeric function. Now, obviously, all of these observations spur the question of whether this multi-step translocation and satellite remodeling mechanism is also observed in human chromosome rearrangements. And this is something that we have begun addressing using the 1421 translocation, which is linked to a special case of trisomy 21 in humans. Taking advantage of an available model cell line that contains this unbalanced translocation. Now, using this cell line, we have carefully mapped the presence of alpha satellites in the, let's call them original and the translocated derivative, as well as identified multiple back clones that come from the centromeric region and pericentromeric region of the original chromosomes and assessed their location in the derivative. This analysis has revealed a very similar scenario to what was observed in cattle, with the derivative chromosome containing an inversion and loss of satellite sequences that can only be explained by a two-step rearrangement model. Of course, all of this begs the exciting question, which is, can we actually map these changes at the sequence level and gain a better insight into their dynamics from this new information? Of course, I don't have to tell you that constitutive heterochromatin has been the purgatory of genome sequencing projects. So in fact, in spite of the 20 years gone by since the draft genome sequence, the GRC 38 reference that we have still has tremendous gaps, specifically affecting 8% of the genome, and most of them located to the constitutive heterochromatin regions. This is a consequence of multiple things, including biases in library preparation, but most of it is a problem emerging from the limitations imposed by sequencing read length when you have to deal with extra long, highly repetitive sequences. And so the kinds of gaps that you still had in the GRC38 include 150 megabase pairs of unknown sequence, and most of them are gaps and lack of information focusing on, yes, pericentromeric, subtelomeric, large satellite arrays, acrocentric chromosome arms, precisely the regions that we are interested in. Of course, in this rapidly evolving field, technology developments keep opening new doors. And in fact, just about 1 month ago, the Telomere to Telomere Consortium has released a preprint with a new genome assembly that provides the most important improvement on the human reference, I would say, since the first genome draft, with a huge increase in the amount of sequence now available. So we've had the opportunity to discuss the approach of the T2T consortium in this recent review, but basically it relies on the implementation of long-read sequencing using both nanopore and PacBio technologies, coupled to the development of sophisticated algorithms for genome assembly, and in parallel, the use of multiple other technologies for correction and validation of the assembly, including things as traditional as in situ hybridization and flow sorting. So an approach, the telomere to telomere sequencing achievement that relies heavily on the use of multiple techniques and technique interdependency. So in partnership with the BioEasy genomics infrastructure, and taking advantage of the strengths of nanopore sequencing technology it has, we have been developing and adapting the T2T principle to derive focused methods for efficient mapping of ROB centromeres. So in this relatively recent project that unfortunately slowed down significantly because of the pandemic, we have taken advantage of the model cell line that I mentioned before that contains the derivative 1421 chromosome to acquire sequencing data either from native DNA and focusing on long-read sequencing and sat-DNA analysis, but also we've attempted to generate focused data on our chromosome of interest through chromosome sorting and sequencing of sorted chromosomes. So for the whole genome sequencing approach, we actually have been able to generate some data sets with approximately 10x coverage of the human genome, of the cell line specific genome, in which most of the reads are over 100 kbs in length. And these results have been analyzed focusing on multiple aspects, but predominantly on the identification and mapping of large tandem arrays, obviously of satellite DNAs, on neighborhood sequence analysis, and on the search for reads that could be mapped simultaneously to chromosome 14 and chromosome 21 in order to identify those that might help us reassemble the ROB translocation. Of course, with the new release of the T2T data, this analysis will be restarted now using the new assembly as reference. However, the ideal approach to understand the structure of the translocated chromosome would actually require that the normal chromosomes aren't present in the same sequencing library. And in fact, part of the success of the T2T project is justified by the use of a haploid hydatiform genome sample, which really simplified a lot the genomic reconstruction. So to achieve this focused sequencing, we have been working hard on establishing methods for chromosome sorting. And these are based on the use of fluorescent dyes that have different affinities for AT and GC sequences. And this plot shows you the typical staining pattern that we have obtained for our model cell line. And as you can see, we are able to discriminate aggregates of chromosomes and even individual chromosomes based on the differential staining of these dyes. And each of these signals can then be sorted to define a specific chromosomal sample. Of course, we then need to identify which chromosomes are present in that sample. And this is done by performing degenerate PCR on each of the samples isolated in order to generate labeled probes for in situ hybridization. Each of these probes are then hybridized to metaphase spreads so that we can know where our chromosomes of interest are. So in our particular scenario, we've found that, well, kind of unfortunately, the 1421 derivative co-segregates with chromosome 9, 10, 11, and 12 because of sequence and content and size similarities, whereas the 14 and the 21 can be isolated independently. And we have performed a pilot sequencing round with approximately 4,000 sorted chromosomes, and I'm showing you here the results of the mapping of these reads, where you can see that in fact our sorting approach seems to have been quite effectively able to isolate the chromosomes of interest, with the 14 and 21 showing up in kind of equivalent amounts. So we think the stage is now set for us to start performing a more in-depth sequencing of samples containing the derivative chromosome independently of the sequencing of the normal 14 and the normal 21 chromosome that will allow us to hopefully be able to generate a detailed map of the satellite regions. So in summary, I've provided you with an overview of what is going on in the field of satellite research, why these DNA sequences are important, how they impact on genome function and dynamics and evolution, which kinds of technologies can we use to map them. And the message I want to leave you with is that we are truly at a very exciting stage for satellite DNA research in particular, given the recent technological developments, and that we believe that with the combination of these developments with more traditional hybridization-based approaches, we are at the perfect timing to be able to start generating robust models of the repetitive genome and its dynamic sequences. And with that, I will finish my presentation and look forward to your questions.